Today we're going to kind of look at mutations, and if you remember when we, back in the first quarter when we were talking about, for example, meiosis and mitosis, we talked about mutations previously with like with like duplication and deletion and inversion and also non-disjunction. So those were chromosomal mutations. Mutations that affected whole chromosomes or whole chunks of chromosomes at a time. And that really went with what we were looking at that point, which was like the macro structure of DNA. But since we've just talked about protein synthesis, and since we've just talked about gene regulation and control, we're looking at the microstructure. And so the mutations that we are looking at today are called gene mutations because they only affect a handful, a number of base pairs, or even one base pair. So we call them gene mutations because they take place on the level of the gene and not on the level of the chromosome. I included this background with the spider because big comic book fan, and a lot of comic book heroes and villains like Spider-Man got their powers because of mutations. You know, a radioactive spider bit Peter Parker and mutated his DNA to have all of those spider-like qualities. Or if you're a Hulk fan, Bruce Banner, the gamma radiation mutated his DNA to have the insane strength of the Hulk. So like I said, today we're looking at gene mutations, which is a change in the nucleotide sequence of a gene. So it doesn't have to do with altering the structure of a chromosome at all, like was the case when we previously talked about mutations. This is just a change in the nucleotide sequence of a given gene. It may even only involve a single nucleotide or a single base pair. It could be that localized. And it may be due to a copying error, so DNA replication or even transcription having an error with it. Could be because of a chemical, a mutagen, that alters the genetic sequence. Could it be the result of a virus? When the virus injects its viral genome into your cells, pieces of it remain and are incorporated in your DNA. Actually, it's thought that at present 10% of our DNA is viral in origin and conserved through our ancestors. So here are, the different types of gene mutations that we're going to talk about. Point mutations, substitutions, insertions, deletions, and then frame shifts. So a point mutation is the change of a single nucleotide. So one base pair is different from what it should have been. Point mutations is a general category that includes an insertion, a deletion, or a substitution of one nucleotide. All right? So point mutation is an umbrella term that includes the more specific types of mutations of deletion, insertion, and substitution when they involve a single nucleotide. Point mutation can give rise to sickle cell disease. It's the result of the substitution of one nucleotide. It occurs in a gene for beta hemoglobin, where a glutamine is replaced by a valine. The hydrophobic or hydrophilic glutamine is replaced by a hydrophobic valine, and that's why you get this sickle shape. Frameshift mutations, on the other hand, are the insertion or deletion of one or more nucleotides. So notice, what's the difference between these frameshift ones and these point mutations? Okay, so a frameshift can occur from a point mutation, from the insertion or deletion of a single nucleotide, but substitutions are point mutations that cannot cause, or the substitution of one nucleotide cannot cause a frame shift. The insertion and deletion of a nucleotide can cause a frame shift. So let's think about this. We've got point mutations that include the changing of a single nucleotide. This includes inserting one nucleotide, deleting one nucleotide, or substituting one nucleotide for the other. When a point mutation is insertion or deletion of one nucleotide, a frame shift results. It changes the reading frame or the sequence of codons, just like changing a sentence. It causes the protein to be built incorrectly because there is substantial damage. So let's take a look. Here is an original sentence made up of three-letter words, just like you can view each codon as a three-letter word. The fat cat ate the wee rat. Now, if we inserted a nucleotide in A, look what happens. The fat cat, we insert an A. Now, codons are still read 3 nucleotides at a time. So this T gets pushed over to the next nucleotide, and the E gets pushed over to the next. And so each codon is now different. We've shifted everything over to the right one. And so when a frame shift mutation occurs, when an insertion or deletion of one nucleotide occurs, it changes the reading frame. And each codon, starting with and after the affected codon, are now different. This is why, and I hope you pause to think about it, this is why a point or mutation that results in a frame shift is much more likely to be deleterious than a substitution. I'll give you a second to think about why that would be. An insertion or deletion is more likely to be serious than a substitution. Right? Because a substitution would only change the codon that substitution took place in. An insertion and deletion would change that codon and all of the consequent codons. So let's look at that same example with an amino acid sequence. Look at the top. Our set of 4 codons and the 4 matching amino acids. If we remove the G, look how it changes all of the amino acids starting with the glutamate and then after. Here is, lastly, just an animation that shows you some different types of gene mutation. Substitution, insertion, deletion, inversion. You can switch around nucleotides as well. All right. That's all I wanted us to look at for today. We'll be talking more about this here in class tomorrow.